All right, hi everyone. Welcome. Thanks for joining us for today's Biocompare webinar, Finding Better Drug Targets, Strategies for Mapping the Cell Surface Proteome, sponsored by Sinobiological and Eppendorf. We're excited to have you with us, what promises to be an engaging and informative session. Today we'll be exploring the latest advances in cell surface proteome profiling and how they're transforming target discovery, especially for cancer therapeutics. I'm Tamlin Oliver, Managing Editor of Biocompare, and I'll be the moderator for this webinar. I'm pleased to introduce our speakers, Fabiana Perna and Arun Wiita, both of whom bring a wealth of expertise in cell surface proteome mapping. Arun is a physician scientist and professor in the Department of Laboratory Medicine and Department of Bioengineering and Therapeutic Biosciences or Sciences at the University of California, San Francisco. His research focuses on mass spec-based proteomics, target discovery, protein engineering, and cellular engineering toward the development of novel immunotherapies for hematologic malignancies, while his clinical duties center on molecular genomic pathology. Fabiana is a physician scientist and senior member in the Department of Blood and Marrow Transplant and Cellular Immunotherapy at Moffitt Cancer Center, where she also serves as medical director of the cell therapy facility. She leads an independent research program and provides clinical care for patients with hematologic malignancies. We're going to start the webinar off with Arun, who will hand off to Fabiana, and then I'll be back for the Q&A. Okay, Arun, let's get started.
Good morning, everybody. Thanks for joining. Bright and early here on the West Coast where I'm calling you from. I'm going to show you some nice pictures of San Francisco in the background of some of my slides. Really happy to be here today to talk about the cell surface proteome. I'm going to kick us off with kind of an introductory talk about how we map the cell surface proteome, thinking particularly about some of the methodologies, pros and cons, overall strategies, why we do it, and then a couple vignettes about some of the work we've done in our lab to discover new therapies and then discover new immunotherapy targets and then going on to actually develop new proof of principle therapies. So really happy to be here. I'll just briefly list my disclosures here. So to kick us off, I'd like to think about we're going to the frontier of the cell. That's where we're really focusing. If we zoom in here on the plasma membrane, particularly in a cancer cell, this is the place where tumors interact with their microenvironment, where growth factors come and engage metabolic programs that promote oncogenesis, where the immune system interactions occur that, say, create immune suppressive microenvironments, as well as we always have this ability to think about this complex interplay, not only between tumor cell proteins and other external microenvironmental factors, but proteins interacting with each other within this crowded lipid membrane environment. And this is a nice simulation from about several years ago. And within this, while there's incredibly complex biology that's occurring at the plasma membrane, we can also think about it as a rich source of therapeutic targets. Actually, about 65% of FDA-approved drugs target plasma membrane proteins. Many of these are small molecules, but as many of us know, the most burgeoning class of cancer therapies are immunotherapeutic agents directed at cell surface proteins. And so if we're really thinking about how we discover new drugs for cancer, we have to think about the cell surface. And the term that we typically use for this mosaic of proteins at the cell surface is going to be the surfaceome. And so that's really what we're focusing on today. And again, for many of you joining, you're already well familiar with this, but just to review briefly, when we are thinking about targeting the surfaceome and individual surface proteins, what are some of the therapeutic modalities that we can engage? And so within this, we actually have several different options of antibody-based therapeutics. And this is actually just a subset of the ones that exist. But we can think about naked monoclonal antibodies, agents such as rituximab, which have been around for decades, Herceptin, that target cell surface antigens, CD20 and HER2 respectively in those examples, that engage endogenous immune cells to recognize and kill tumors via that initial recognition of a cell surface protein that's upregulated on the tumor cell. We also have newer modalities that are incredibly expanding, such as bispecific T-cell engaging antibodies, where you have a dual functional antibody where one side binds to that tumor surface protein, the other side binds to CD3 on a T-cell and creates an artificial synapse that leads the T-cell to kill the tumor. And you also have modalities such as antibody drug conjugates or similarly radioimmunotherapeutics, where you can conjugate a cytotoxic warhead to an antibody that then recognizes the surface antigen, is internalized into the tumor cell, and then enacts a cytotoxic program to kill the tumor. So we have those options. And then also we have as well engineered immune cells. So the largest of this category are definitely CAR T cells, or chimeric antigen receptor T cells. I don't have time to go into incredible detail about these for those who aren't as familiar, but the key thing to know is both for CAR T cells as well as CAR NK cells, we can take a patient's own immune cells, uh, introduce a fusion gene that is a fusion between typically an antibody fragment or some other recognition element that's going to recognize that surface protein in the tumor cell. And then inside the immune cell, we enact the downstream cytotoxic signaling that enables this reprogrammed immune cell to kill the tumor cell as a function of that surface protein recognition. And so these are incredibly powerful modalities. We have FDA-approved therapies that fit into all these classes that I'm mentioning here. But the issue is within this, sorry, my animation was a little funky there, but enable actually therapeutic development for these surface antigen targeted strategies, we need a good surface antigen. And so how do we define that? So to dig into that a little bit more, the way I like to think about it in my group is there's a few criteria that we would find for our ideal surface immunotherapy target. And so one of the first ones I like to think about is ideally we have a target that's expressed at high abundance on tumor cells with minimal intertumoral or minimal, as well as minimal intratumoral heterogeneity. And so what do we mean by that? So when we talk about intertumoral heterogeneity, that means ideally this target is expressed on the tumors from all different patients. It's not only highly expressed on 5% or 10% of patients. We're developing a therapy that can benefit all the patients with that type of cancer indication, as well as intratumoral heterogeneity. That's very important because ideally all the tumor cells in an individual patient's tumor express the target. Because the problem is if you have a subset of tumor cells that are negative for that antigen, they can easily grow out and relapse. Even if you kill the 90% of tumor cells that express the antigen, those 10% can be the nidus of relapse cancer. So that's what we want to avoid. So those are some of the criteria initially for the target. And in terms of the actual density of the target at the cell surface, in terms of absolute number of protein copies per cell, that can also be really important in terms of thinking about how can we access this therapeutically. CAR T cells, bispecific antibodies, tend to be very sensitive, down to potentially low thousands, sometimes even hundreds of copy numbers of antigens per cell. Antibody drug conjugates, for example, often might need antigens in the high tens of thousands to hundreds of thousands of copy number per cell density to be effective. So that absolute number of protein copies per cell is also important criteria. But in general, we want things that are fairly highly expressed. And then a really important concept to consider is we need these antigens expressed at low density or ideally absent on normal essential tissues. And the reason this is critical is there's the concept of on-target off-tumor toxicity, where we can develop a highly specific therapy, say based on a highly specific antibody, but the issue is even if it doesn't have any off-target activity, if that target is expressed in normal essential tissues, you can lead to unacceptable side effects. And sometimes, in some cases, this has even led to patient deaths in clinical trials because of this on-target off-tumor toxicity. So ideally, finding targets that are as clean as possible in terms of only being expressed on tumor cells, not expressed in normal essential tissues, is something that we really think about a lot. And how do we thread that needle of a therapeutic index? Some additional criteria I think that's worth mentioning. Again, I already talked about that the CAR Ts and TCEs are more sensitive to low antigen density. So these are, for these targets, for those modalities, those are ones we have to consider more about these toxicity issues. So you may find a target, though, that might say you have a target that is very high on tumor cells, low to moderate on normal tissues. Maybe that's something you want to develop an ADC against because you might have a better therapeutic index, but not a T cell engager because that might have too much toxicity. So these are decisions you can make depending on the target that you discover. And then what I like to think about are other nice to haves in development. Things like tumor genetic dependency, where the tumor can't lose the antigen as a resistance mechanism and still survive because it's providing some critical signaling to the tumor. Again, not necessary. CD20, we've used that for decades. We have no idea what it does biologically, but it's a great target. And so you don't necessarily need this. It's a nice to have, not a need to have. Similarly, thinking about minimal circulating proteins, say cleaved forms of a protein that can form a sink for your administered therapy. Some therapies, this makes a difference. Some it doesn't. It's hard to know a priori. Think about low isoform complexity. There are different splice forms or other proteoforms of this protein where some may be bound by your antibody fragments, some may not be. This may be a useful thing to think about, or their cleave forms, but it doesn't always make a huge difference. And then also one of the considerations is a target from a structural perspective easy to develop an antibody against? Does it have a well-formed, large, recombinantly expressible extracellular domain that's a globular domain, easier to put into antibody development pipelines, rather than something like a 7-pass transmembrane GPCR with very small extracellular loops that you can still develop something to target that is just harder. And so these are all criteria you can use to think about how do you discover and work up, validate a new target. And so getting into this, how do we find these new targets, right? And so there are different ways to do this. Historically, people have done this through hypothesis-driven approaches, flow cytometry, understanding basic biology, then moving into large-scale transcriptional-based screens. The problem with using RNA as a target discovery strategy is that in many cases, the abundance of mRNA in a cell does not well predict the abundance of an actual protein at the surface. And so the mantra of my lab at UCSF is biology happens at the protein level. Let's look at the proteins. And so the way that we choose to do this is using cell surface proteomics. And I'm going to dig more into this in the next few slides in terms of specific methodologies. But as a general strategy, most of these approaches are going to be an integration of chemical biology and mass spectrometry. We're going to be using chemical strategies to decorate cell surface proteins, typically with a biotin handle, ideally using cell membrane impermeant small molecules that catalyze this reaction. And so the reason we do that is because that way we can selectively focus on the cell surface proteome. Using that chemical handle of biotinylation, we can incubate with streptavidin-coated beads. That interaction between streptavidin and biotin is one of the strongest non-covalent interactions that's known, and this is a common strategy in chemoproteomics and many other forms of biochemistry. So that enables us to pull down our biotinylated cell surface proteins on those streptavidin beads, then use very harsh washes using things like guanidinium or urea, for example, that will denature, wash away, ideally, all those intracellular proteins, and then we'll have our set of enriched biotinylated cell surface proteins on those beads, and then we can use different methods of enzymatic and protease treatment, depending on exactly which workflow you use, to prepare peptides that we can load into the mass spectrometer. And I'll talk a little bit about in terms of the mass spectrometry methods. But the key thing to know is in the end, is typically what we'll find is somewhere in the ballpark, and we've applied this both to cell lines as well as primary patient samples, we'll often get somewhere in the ballpark about 600 to 1,000 cell surface proteins that are both identified and quantified by mass spectrometry. And some literature suggests any individual cell may express in the ballpark of 2,000 to 2,500 unique cell surface proteins. So I'm not claiming we're seeing everything in these experiments, but we're seeing a large set. And there probably is a bias toward more abundant proteins are what we're detecting. And so in that case, those are probably the ones more useful for immunotherapeutic targeting anyway. The really low abundance things are going to be very hard to go after. So we're probably seeing the things that we want to see for the most part using this methodology. And again, we're good doing this directly at the protein level. So that's really informative. And so to get a little bit more in this introductory talk about what are some of the different methods. And so I'm highlighting here a really beautiful review written by my really close colleagues at UCSF, Kevin Leung and Jim Wells. I can refer you all to it if you want to get more detail on this, but really talking about the different methods for how to do cell surface proteomics on that technical level. And so what they highlighted in the review, and I agree with this, is the main structure of how you can think about cell surface proteomics. The oldest version of this is using traditional biochemical methods of density gradient ultracentrifugation, or now there are kit-based methods for membrane isolation, essentially. This uses established biochemistry, is a big advantage. You don't necessarily need to use new molecules, new protocols. This may be more applicable, we found, say, for solid tumor samples, basically, actually primary tissue specimens. This actually potentially can be more effective because you don't need to dissociate tissues ahead of time. But the issue is that they tend not to be very pure. There's still a lot of impurities, whether from intracellular membranes, other background that comes across. Using the better method of ultracentrifugation, basically access to an ultracentrifuge, not everyone has that. So these are accessible, but maybe not always the best for every experiment, and they can get pretty complicated when you're using ultracentrifuge-based methods. What we've used for the most part are chemical labeling of extracellular proteins. And so one of the most common methods is methodologies that's existed for a long time of surface biotinylation using an NHS-based chemistry, using this sulfo-NHS biotin probe. It's cell impermeant, potentially can be released from beads via a disulfide reduction step. This type of approach is very accessible. There's newer approaches using biotin tyramide-based labeling, whether via HRP or Apex, that have also been published as well. These can be quite rapid, which is good. They're easy to perform. The protocols aren't that hard. The only drawback is you do still get a fair amount of intracellular labeling with both of these approaches. The sulfo-NHS biotin is nominally cell impermeant, but we still see a lot of background. It really labels lysines quite efficiently, and it can still get into those background labeling with cells. So that's one drawback, but we still use these methodologies to complement. Our most frequent method that we use is the so-called cell surface capture method, initially described by Bert Volscheid's lab at ETH Zurich. And this creative method essentially labels cell surface glycosylated proteins by gentle oxidation of vicinal diols on glycans. After creating that aldehyde, you can come in with a biosite and hydrazide reagent that will covalently link a biotin to those sugar residues. And so what this does is, given that 85% of the cell surface proteome is thought to be glycosylated, you can actually get more specificity for cell surface proteins. You get much less intracellular background, and you can do this pull down. The labeling is a bit longer to do. It's a longer protocol, but it probably is more specific. And you do have the caveat, though, that you may miss that set of non-glycosylated proteins. But this is probably our workhorse in the lab. And then finally, I'm not going to talk more about, but there is another area of cell surface proteomics using antibody-based methods, whether that's a readout by CyTOF or a readout by next-generation sequencing using approaches like CytSeq. Again, I'm not going to get into this too much. These techniques are useful because they can get you single cell information on cell surface proteins, which is great. The big limitation is the biggest panels might be 200-300 antibodies, but they have to be already known targets, have antibodies a priori. You're not going to do new target discovery this way for the most part. It's going to be useful for studying biology of known targets at the single cell level, because all the other methods I mentioned to you, a drawback are they are in bulk, but we don't typically think about this as a good target discovery method, but it's really useful for validation and thinking about intratumoral heterogeneity, for example. Okay. And so one other technique that just really is emerging, I thought was really interesting, just to mention really briefly, and this is not an official endorsement by any means, but there is a new technology that integrates spatial proteomics with cell surface proteomics using SynCell's microscope that can actually use photocatalytic labeling of plasma membranes and then enrich out of fields of vision if you have a tissue sample, and then do cell surface proteomics that way using downstream mass spectrometry. So spatial proteomics, an emerging field we're not going to talk about today, but I think an exciting development just to think about going forward. When it comes to actual mass spec, I think it's important to say there are many different options. This is not a mass spec talk. We don't have time. I teach a whole graduate class on how to do this. And so, but just to say really briefly, there are many different methods of quantitative mass spectrometry. The one that we favor for cell surface proteomics now, that we think is the most robust, reproducible, applicable primary sample cell lines, is data-independent acquisition, or DIA. This can be done either on Bruker or timsTOF-based instruments or Thermo Orbitrap-based instruments. If you'd like to learn more about that, we recently published a short methods paper on doing surface omics using DIA proteomics. So I'll refer you to that if you'd like to check it out. Okay, so then now that we acquire this data, how do we actually select a good target? So I'm going to spend the next couple minutes talking about this, how we actually do target filtration. So I think a really important thing to say is there's not a standardized approach in this field. There are many different resources available, many different bioinformatic pipelines that can be developed. Different people do this in different ways, and you may want to prioritize different approaches depending on your specific indication. And so what are some of the limitations here? When we think about on-target off-tumor toxicity, we don't have a great public resource of normal tissue cell surface proteome. And so what that means is that we're comparing to transcriptional data, which again, we know doesn't always translate into what's actually at the cell surface. So that's a limitation, but it's the best we have. But this is still really critical in terms of toxicity assessment. So I think that's a really important limitation to acknowledge. Um, Again, this is more like a resource slide. I'm not going to talk too much about these in detail, but to say there are several databases that can be used to both predict whether a protein is at the cell surface, what's its topology, like which domains do we think are extracellular, which amino acid sequences. That can be really important in terms of thinking about your drug development downstream. So there are many that are existing. Just to point them out, this Uniprod is probably a looser filter. Many things might be annotated as potentially plasma membrane, not experimentally validated. This in silico surface, human surfaceome from the Volshade lab. This we think is a very strict filter for cell surface proteins that meet quite stringent primary sequence criteria, but it may have many false negatives as well. And so there's different databases. I'm listing them here. You can check them out yourself. In terms of resources for actually thinking about target discovery, what we consider the single most comprehensive resource to evaluate target expression is the Human Protein Atlas. And many of you may have used this resource already, but it really is impressive work led by Matthias Uhlin in Sweden, as well as Emma Lundberg, who's at Stanford. They've done a beautiful job integrating tons of different multi-omic resources and a ton of their own data defining mostly antibody-based immunohistochemistry for ideally the entire proteome. The quality varies from antibody to antibody, but they've integrated many other people's data sets from mass spectrometry, serum proteomics, single-cell RNA-seq. And so this is a really comprehensive, beautiful resource that we really take as our initial one-stop shop to assess the quality of a target in terms of expression on normal tissues, distribution on the tumor samples that they have in their database. And then there are additional databases that you can think about that we use frequently for more detailed downstream assessments. DepMap, large-scale CRISPR screens to look at genetic dependency, as I mentioned. BloodSpot, we can look at in terms of expression of targets on hematopoietic and stem and progenitor cells, which is important for hematologic malignancy target discovery. And then the human proteome map is large-scale shotgun proteomics of normal tissues that can also be useful. There are others out there as well, but these are some that we think about. Okay, and then just highlighting some of the features in Human Protein Atlas that are most useful, I think, in terms of the tissue IHC, single-cell RNA-seq, cancer data sets, circulating fractions, as well as cell line expression. Okay, and so the last 2 minutes, what I'm just going to talk about is a few examples from my lab very briefly. I don't have time to go through this, to say how we've used some of these strategies to find new targets. And so several years ago now, Matt Nix in my lab applied cell surface proteomics to try to find new targets for this very poor genetic subtype of B-cell acute lymphoblastic leukemia. We discovered a new target called CD72 we think is highly promising in the space. We're about to start a phase 1 trial of CAR T cells we developed on this. I didn't have time here to talk about the protein engineering we love to do in the lab, but in this case, we demonstrated that we could take this target and use for the first time a fully computationally designed synthetic nanobody yeast display library to select highly selective and specific nanobodies against CD72, put them into a CAR T cell, create highly efficacious CAR Ts that, again, we're trying to bring to the clinic. As another example, we did a large-scale study of the cell surface proteome in multiple myeloma. We identified a target called CCR10 that we think is very promising, had never been identified before. And so in this case now, what we've now done is here using protein engineering for a 7-pass transmembrane GPCR, we actually used AlphaFold based modeling and computational structural modeling to identify we could enhance through amino acid mutagenesis the natural ligand of this target CCR10 called CCL27. And by mutating amino acids within this pocket, create a much more effective CAR T cell based on this natural ligand. And so this is up on bioarchive if anyone wants to check that out. And then finally, the last thing I'll mention very briefly is a big emphasis in my lab now is thinking, can we go beyond just target antigen expression levels? The proteomics I told you about is focused on expression. Can we go past that? And so one example that we published is a technology we call structural surfaceomics. We use another chemical biology strategy of homobifunctional chemical cross-linkers to lock proteins in situ in their native conformation at the cell surface, then do our cell surface biotinylation. And then we've developed software tools that allow us to map back all of these cross-links that we identify to either PDB published structures or AlphaFold predicted structures, predict unique tumor-specific protein conformations, that three-dimensional shape that can be targeted. We found an exciting target, acute myeloid leukemia, made highly efficacious CAR T cells. But to say the cool thing is once you find a target, you make different modalities. And so now we actually turned that fully human antibody we developed via Fab phage display that's conformation selective, we turned it into a radioimmunotherapy working with Rod Flavella's lab here at UCSF. And so we found these to also be highly efficacious in AML tumor cells. And we're also trying to push this toward the clinic as well as a treatment for acute myeloid leukemia. So with that, my time is up. I'd just like to say very briefly that I really think surface proteomics is a very powerful tool for immunotherapy target discovery. As I mentioned, there are different ways to perform surface proteomics and multiple ways to analyze the data. There's not a single pipeline. So you have to do work. Finding the target is half the battle. After that, what we love to do is how do we think about the protein engineering to make a new therapy to go after that? That's actually where a lot of the fun comes in. And then I really think the future is trying to integrate proteomics, chemical biology, and computational methodologies. I shouldn't say just CAR targets, but immunotherapy targets in general, beyond just protein abundance or antigen expression. So with that, my thank you got misaligned here, but say thank you all for your attention. I mentioned many of our folks in the lab along the way, our collaborators, funding sources. Thank patients and their families for participating in research. And with that, I will hand it over to Fabiana.
Thank you so much, Arun. Wonderful talk, and I'm very happy to be here and share what my lab is doing to map the cancer surface proteome with the overall goal of understanding and treat hematological malignancies. I will start from a clinical question, so a clinical need, and I will give you two examples. One is on multiple myeloma and one is on acute myeloid leukemia. So, in multiple myeloma, immunotherapy is really changing the treatment approaches. It is a highly effective, however, this disease remains not curable. Many patients still relapse. One of the mechanisms that Dr. Wiita mentioned is antigen escape. And this highlights the need of identifying novel targets that can bypass some of the resistance, which is mainly associated with the one molecule, BCMA, which represents the most important immunotherapeutic target for multiple myeloma. A second challenge in the field is posed by tumor heterogeneity, which is which both is due to the pre-existence of clones that are BCMA low or BCMA negative, as well as to the downregulation of the target antigen under immune therapeutic pressure. Also, current immunotherapies associate with significant toxicities, which represent a limitation in the field of immunotherapy in general, especially CAR T cell therapy, which is the main focus of my research lab. And so, as Dr. Wiita was rightly mentioning, there are issues related to on-target off-tumor toxicity when the target is expressed on normal tissues, as well as other types of toxicities, such as cytokine release syndrome of ICANS or ICANS in the case of immune-based therapies, especially CAR T cell therapy, and my lab has been pretty active on this aspect as well. So all these clinical challenges highlight the need of novel targeting platforms that can recognize novel target antigens expressed on the surface proteome of cancer cells, with the overall goal of bypass resistant mechanisms, broaden eligibility, and improve long-term disease control. So, in order to map the surface proteome in search of novel target antigens, we also face some technical challenges, which are mainly represented by the fact that plasma membrane proteins are not abundant as cytoplasmic proteins, they are highly hydrophobic and heavily post-translationally modified. And so, as I mentioned in prior slides, we also use enrichment methods, so primarily based on biotinylation, so labeling of the cell surface proteins, followed by advanced mass spectrometry. There is also the challenge posed by the fact that there are few computational tools that annotate cell surface molecule expression. And so I'm sure many of you deal with the large data sets of RNA-seq or mass spec data. So how do you recognize, how do you select molecules that are likely expressed on the plasma membrane other than the common cluster of differentiation molecules? And so in that respect, my lab has been trying to develop some comprehensive data sets in order to annotate cell surface molecule expression. And finally, as we were just saying, tumor heterogeneity, so this drives also diversity in the surface proteome. And so my lab is trying to focus on specific disease states such as relapse post-immunotherapy to see what are the changes that occur at the cell surface proteome, and also leveraging our understanding of the disease biology to inform more sophisticated search of potential novel target antigens. So I'll give you some examples that we are performing. We did some mapping of the cell surface proteome of multiple myeloma. So we label, as we just said, the surface proteome with biotinylation. We used a panel of 6 multiple myeloma cell lines bearing high risk genetic features. And through that, we generated a pool of potential candidates. So we also integrated this data with RNA-seq from over 900 patients to identify molecules that are also relevant in the patient population. And so to narrow down the focus to molecules that are commonly overexpressed at the protein level in cell lines, as well as at the RNA level in patient samples. And we develop what you see in the middle of this slide, a comprehensive data set for cell surface molecule annotation, which includes 5 distinct repositories that were mentioned by Dr. Wiita, and this creates a surface scoring. So if a molecule is included in at least, let's say, 2 of those data sets, there is a higher chance of being localized on the plasma membrane compared to a molecule that is not included in any. And each one of them is based on different methodologies, such as transmembrane prediction, machine learning, antibody-based, and so on. So what we found was that on multiple myeloma, there were 3 main protein networks, which reflects also the disease biology. So not only we look for molecules that can be targetable, but also these molecules actually contribute to disease initiation and progression. So we found the molecules involved in adhesion. So as you know, cancer cells can attach to the stroma, transporters that can promote survival of cancer cells, and also molecules involved in immune related mechanisms. So basically we chose our own criteria to narrow down the list of selected proteins, starting from more than 4,000 molecules. And so we identified about 300 that were commonly expressed in cell lines and patients, to about 200 were primarily related functionally to immune mechanisms. And then we spent extensive time in validating by Western blot or flow cytometry, depending on commercially available reagents, to look at the expression of these molecules both in primary samples from patients as well as in normal tissues. So from that we identified some targets associated with multiple myeloma. When we look at the expression of our top candidates in a large institutional data sets of patients with relapsed multiple myeloma, over 800 patients, we found that in particular two of them were more highly expressed. And these seem also to impact clinical outcome. So with the ILT3, which was previously described primarily in AML, it's a negative immune regulator of T cell proliferation. We found that this seems to occur also in multiple myeloma because of the genetic targeting of this molecule would increase actually T cell proliferation in co-culture assays. And so from that, we developed a bispecific T cell engager that recognizes this molecule of multiple myeloma as well as a CD3 on T cells, bringing the immune cells in proximity of the cancer cell, promoting the killing. And so we ran several experiments to show the efficacy of this approach. More recently, we also I was telling you, we try to focus on specific disease state. So, in order to address the issue of relapse post BCMA CAR therapy, we had the opportunity to analyze a unique cohort of bone marrow samples from patients who relapsed after BCMA CAR therapy. And what we did was a precise measurement of antigen density. So, in addition to just look at the expression of this clinically used target, which is BCMA, we also quantify the number of molecules per cell. And this is an important information which is increasingly becoming relevant, especially in the field of CAR therapy, because there is a threshold that is required for optimal CAR T cell activation. And so what we found is that in this cohort of relapsed patient, even if the cells remain positive for the target antigen, however, the antigen density was below that threshold that is required for optimal CAR activation, so suggesting that this could contribute to immune escape. And so we look at the expression of our targets that we had identified to see whether in this cohort of patients, perhaps we can leverage the expression of another target to rescue these relapses. We found the SIMA4A was highly expressed. We initially used the CRISPR knockout cell lines to see whether this molecule had an impact in the disease biology, which is one of the nice to have features that Dr. Wiita mentioned. And we indeed found that when we knock out this molecule, the proliferation of the cells, of the myeloma cells, goes down in vitro. We also set up an assay for migration, where basically there are two chambers. The top chamber basically has the cells without FBS, and so normal cells tend to migrate to the lower chamber where there is FBS. But in the case of the SIMA4A knockout cells, actually the cells are impaired. So it seems that this molecule impacts also migration. In vivo, we found that when we inject knockout cells in NSG immunocompromised mice, actually mice can survive much longer, and we measured the tumor burden with measuring the human kappa or lambda chain depending on the cell lines that we used in the mouse serum. So based on these initial features, we found that, for instance, this is an example, this molecule was highly expressed in tumor cells, has basically a function in the disease biology. And so we move forward in constructing some novel CAR T cells by developing the first novel monoclonal purified antibodies recognizing the extracellular domain of these molecules. And we screen several antibodies, so from which we constructed CAR T cells. Then we screen CAR T cells in vitro, and we found that actually 5 of the initial pool of CAR constructs were more effective in CTL assays in vitro while maintaining specificity, so they were not killing, for instance, the SIMA4A knockout cells. But the real issue is really the expression on normal tissues. Because while it is relatively established how to access the efficacy and the killing of target cells once you find these molecules to be highly expressed on tumors, the challenge is how to predict potential toxicity of these novel therapeutic strategies. And this is a challenge in the field because also in terms of in vivo models, I mean, we all generally use xenograft mouse models, so it is there isn't basically an established system to address that. So what we did was first we integrated three repositories: the Human Protein Map, the Human Protein Atlas, and the Proteomic database, which provide a simultaneous annotation of protein RNA and protein expression in a large panel of normal tissues. And so by doing this, we compared several immunotherapeutic targets in multiple myeloma that are either in clinical or preclinical use to basically see whether the profile in normal tissues was favorable, at least more favorable than other targets that are currently used. And that was the case for our molecule of interest. We also were able to collect normal tissues, and so we went on experimental protein validation of the expression on normal tissues. And so we used not only the commercially available antibody, but also the novel antibodies that we had generated. And so we look at the expression in by multiplex immunofluorescence and confocal microscopy in the tissues where there was some kind of potential low expression of the molecule. And on the right, you see the positive control. These are not only myeloma cell lines, but primary bone marrow biopsies from 2 patients where the control is represented by C138. And so you see that both our novel binder as well as the SIMA4A commercial antibody were highly expressed, while there was a minimal expression on the normal setting. But we took a step further, and so we also measured by flow cytometry, which is a more sensitive assay, the expression in normal tissues. So this was done in fresh lung cells, where basically the positive control is represented by EpCAM positive cells. And so when we look at the CD45 negative EpCAM positive cells, the expression of SIMA4A was negative, suggesting that the expression does not occur on epithelial cells. Also, we were able to collect several frozen normal tissues, and so we, in a similar fashion, we look at the expression of these molecules. And when we take the commercial antibody, which is more sensitive against this target, we also look at the antigen density in normal tissues. And so what we find is that this analysis suggests that there is a low expression outside the normal monocytes, but still normal monocytes still bear a lower antigen density compared to myeloma cells, suggesting the existence of a therapeutic window. An additional experiment that we integrated in the analysis in normal tissues to address really the toxicity concerns and preparing for a phase 1 clinical trial was the development of a humanized mouse model, which was basically, as is based on the injection of CD34 normal CD34 hematopoietic stem progenitor cells that we derive from cord blood. And so these mice recapitulate human hematopoiesis, and we also injected these mice with the myeloma cells, and then we treated these mice with either BCMA CARs, which are more established, or our novel CARs. And in this way, we were able to demonstrate that the anti-myeloma efficacy occurs without any additional hematopoietic toxicity compared to a clinically validated CAR. I told you at the beginning that one of the disease state we were interested was the relapse after BCMA CAR therapy. And so we tried to model this system in vitro and in vivo by generating a panel of engineered cell lines where we knock out BCMA and then we re-express the BCMA at low levels, mimicking the expression levels that we observed in patient samples. And so what we found is that actually, if you see in this panel C, when we use the BCMA low positive, the BCMA cars, which are represented by this yellow line, actually perform less well compared to the setting where BCMA expression is higher. So really the expression levels impact dramatically the performance of CAR T cells. And so in this setting, one way to overcome this is by basically redirecting the CARs against another target. And indeed, if we use in our case, SIMA4A CARs, we were able actually to control the disease. And this was also performed actually in vivo using NSG immunocompromised mice. So this is what we have done in the multiple myeloma, but with the few minutes that are available on my talk, I also would like to present some preliminary data that leverage the disease basically molecular mechanisms to identify different type of targets compared to the tumor-associated antigens that I explained so far. So in acute myeloid leukemia, this is another heme malignancy, which is very aggressive, associated with a very poor prognosis, and the only curative approach is represented by allogeneic stem cell transplant, which is based on the cytotoxicity of donor immune cells on leukemia, an effect that is called graft versus leukemia effect. However, the transplant is not possible for all patients for reasons of eligibility, and also relapse remains a major cause of disease. So also in this disease, there is a high and urgent need to identify novel targets in order to develop novel therapies that are based on immune approaches. Neoantigens are, I would say, is likely excluded as a target type because the number of mutations in this disease is minimal, especially compared to solid tumors like melanoma or lung carcinoma, which have a very high number of somatic mutations. And indeed, we know that checkpoint blockade works very well in these settings. But in the case of heme malignancies, especially AML, the number of mutations in de novo disease is around 13 mutations. And so this makes the leukemic cells very similar to normal cells, and it is hard for the immune system to recognize potential neoantigens, and this reflects also less successful response to immune checkpoint blockade. So as a fellow, I tried to address this issue by identifying novel leukemia-associated antigens in a similar fashion to what I described before for multiple myeloma. So basically we label the cell surface of a larger panel of leukemia cell lines to map the surface proteome. We integrated this data with RNA-seq from leukemia patients, and we tried also to annotate the expression in normal tissues by integrating the Human Protein Atlas, proteomics database, and the human proteome. And then on a subset of most promising targets, we performed flow cytometry. However, as of right now, even if there are many molecules, especially CD33, CD123, and CLL1, which are in clinical use for AML CAR therapy, there is no FDA-approved product. And the reason for that is that the results in terms of efficacy are quite modest, and there is a massive toxicity because these molecules are generally also expressed on normal hematopoietic cells. In search of novel tumor antigens, we tried as a lab to focus on a new category, which is represented by unconventional neoantigens or aberrantly expressed tumor-specific antigens, which are potentially cancer-specific, but their identification is more complex. So basically, instead of deriving from mutated areas of the genome, they actually derive from alternative splicing. And in AML, even if there is a small number of mutations, however, there is a frequent recurrence presence of mutations impacting splicing factors or epigenetic modifiers. And so we developed a pipeline which is specifically focused on the identification of novel and unannotated isoforms that can contain specific domains that derive from aberrant mRNA splicing. And then we try to prioritize isoforms that are involved in an isoform switch, so that are upregulated at the expense of the full length and most dominant form, because these might suggest biological relevance. And also, we try to prioritize isoforms that derive from genes coding for cell surface proteins. And then we look at the distribution across actually different patients. So we initially applied this pipeline to some genetic models. So again, trying to focus on the biology of the disease when we look at the cell surface proteome. And so we used the cord blood derived normal CD34 cells that were virally infected to express some of these frequent co-expressing mutations such as SRSF2 and IDH2. And we identified some targets. This is an example. So in the top panel, you see the full length protein. At the bottom, a predicted protein structure of protein domain of these isoforms. The green boxes represent potential unique sequences that derive from aberrant alternative splicing. And so by doing this, we are in the process of characterizing the leukemic cells that are expressing these variants. So I will conclude because of time, saying that targeting tumor antigens, as all of you know, is a very promising treatment for cancer. The biology and basic studies suggest that this is primarily driven by accumulation of genetic and epigenetic aberrations, and as they generate transcriptional and proteomic diversity, we expect this diversity to impact also the cancer cell surface proteome, creating basically novel mediators of disease initiation and progression, but also potential novel targets for immunotherapy. As I told you before, one of the critical issues is really the annotation in normal tissues in order to identify good targets that are absent or at least minimally expressed on vital normal tissues. And with that, I would like to, of course, acknowledge my lab, the funding sources, the collaborators, and I think it is about time to get your questions. And again, thank you so much for joining this webinar today. Thank you.
Thank you, Fabiana, for that wonderful presentation, and Arun for yours as well. Those were great, very informative. We're going to start, Arun, with questions for you. Are you ready?
Sure. And I think this would be a good one for Fabiana too, but I can go first. And then I think Fabiana has some good thoughts on this as well.
Oh, wonderful. Thank you. Can you discuss more about which surface proteomic methodology your lab uses most frequently?
Yeah, for sure. So yeah, maybe I'll tackle it first, and then Fabiana can speak to it. So for us, really that cell surface capture methodology of essentially the cell surface glycoprotein oxidation is our workhorse that we use most frequently. But we do like to think about typically like one story I didn't talk about now, and again, I really like Fabiana. I really liked your story about the alternative splicing and new antigens in AML. We have a story we're just about to submit where we're trying to find also aberrantly trafficked proteins to the surface in AML. And to try to validate this, we actually will apply multiple different methods of cell surface proteomes. And often that's what we'll try and do is, in that case, we actually did primary AML surface omics on about 60 primary AML tumors. But for a subset for that large panel, we did the cell surface capture of glycoprotein oxidation, cell surface proteomes. Then for a subset to try to do additional validation, we also applied the NHS biotin type approach. For other strategies, we've used a Wiita agglutinin fused to HRP because it's more rapid, can use smaller sample inputs, for example. So that cell surface capture, the drawback is typically you'll want to have at least a couple million, ideally 10 million cells input. That can be hard for primary samples. Some of these other methods may be less specific, but you can go with less input. And then what we've done for tissue samples, oftentimes like intact tissues, membrane protein isolation, we have found to be more effective because we're always worried about dissociating tissues. And then the challenge is, have you left a scar on the cell surface proteome that's somehow altering it in a way that's going to lead to artifacts? And so that's actually the method that we use for tissues. And we don't do a ton of tissues in heme malignancy. It's a nice thing is the tumors are in suspension for the most part, but that's typically the strategy we use.
Fabiana, did you want to weigh in on that, or?
Yeah, I mean, I share basically a similar approach. So we tend to most commonly use surface biotinylation. I think the decision is very much dependent on the type of samples because with cell lines, we have more options because of the number of cells. But primary samples are, of course, the most precious and the most relevant ones. However, there is a large variability in the quality of the samples, in the number of cancer cells. They don't grow very well in vitro. And so this limits a little bit the possibilities. But yeah, so basically, this is what we also do in our lab as well.
Okay, thank you. I'm just going to skip around on the questions a bit, but Arun, this one's also for you. Are there ways to isolate and probe tumor cells after a primary treatment, for example, CAR T, to interrogate what new cell surface targets are upregulated during antigen spreading as a tumor tries to escape the primary treatment?
Yeah, again, I think I'll talk about it. Fabiana's semaphore story is very relevant as this to well. So just to say, I mean, I think it's definitely feasible. I think the main challenge essentially is getting those samples on treatment, right? So that's the main limitation as we're talking about primary samples is from a clinical perspective, how many of those, again, for heme malignancies, what bone marrow biopsies can you get kind of in a longitudinal fashion during treatment, and then looking at this evolve strategy. Again, going back to the AML story I didn't talk about, in that case, we looked at, 20 some new diagnosis AML as 20 some relapse patients, but they weren't all paired. So we can make a comparison between relapsed AML, but, I think, Trey, it's a great question in terms of could you do this like really kind of in real time? But it's just challenging to get the samples. But yeah, Fabian, your thoughts?
Yeah, I mean, plasma membrane proteins have dynamic expression. So what we do is like we take a static picture in a specific moment. However, they undergo a modulation in terms of expression. So what we did for the semaphore story, we basically were able on a limited number of samples to collect the bone marrow before CAR T cells and after CAR T cells. And that was in our possibilities the best to address basically this question. I think ideally it would be nice to have paired samples that are followed longitudinally to see how this picture changes through time and through basically the various treatment. In our case, as I was saying, so from 6 patients, because we analyzed a cohort of more than 40 samples post-relapse to see what happens after CAR T cell therapy, but only in 6 samples we were able to exactly measure the antigen density before and after CAR T cell treatment. And we do see differences. The most obvious was basically the downregulation of BCMA antigen density, but I would assume that we didn't perform unbiased basically mass spectrometry to look at comprehensively all changes occurring upon BCMA treatment. However, with the selected number of targets that we wanted to basically study, we found that there was a change, there were alterations in the surface proteome after CAR therapy. So the question is very well taken.
Thanks, Fabiana. Unfortunately, we're out of time. So if you submitted a question and didn't get to it, we can try to answer it via email later. But thank you so much, Fabiana and Arun, for the contributing to the webinar today. And also a special thank you to our sponsors, Sinobiological and Eppendorf, for sponsoring today's event. Thank you so much for joining us today, and we look forward to seeing you at our next event. Bye for now.
Bye-bye.
Thanks, everyone.